Hello, everybody, and welcome to my presentation on our vector platform for helper virus-free AAV production with stable CAP and HEK293 producer cells. As you all know, the cell and gene therapies are currently developing at an amazing speed, and so are the associated delivery tools. And when we look at the vectors that are in use, it's apparent that the majority of vectors that is used to deliver the gene cargo to patients for in vivo gene therapy is actually based on adeno-associated virus. And some of the reasons for that are that AAV is a very safe viral vector. It's small and stable, and also an important reason for its frequent use is that it comes in many different serotypes, with additional new serotypes being developed, which all show different tropisms, which means it can be used to target specific tissues in the human body. And this has been a very successful tool, but it's also facing potential problems, in particular for future therapies when we look at manufacturing. So the therapies that are currently in the market, like for example Luxturna, also Gensma, they target ultra-rare, very rare diseases and usually have to be administered in low doses. That means the required product, so the actual viral particles required per year, is very moderate. So we're talking about 10 to the 12th to 10 to the 13th viral particles per year. And for this reason, it has been very feasible and possible to actually produce these viral vectors with the classical production methods as they were originally developed in the universities where the therapies were invented. And these classical production methods usually or very often employ transient transfection of 3 different plasmids, the so-called triple transfection, very often still using even adherently growing cells on cell stacks. However, when we now look at therapies that are currently in clinical development, targeting common diseases like Parkinson's, Alzheimer's, for example, and also therapies that might have to be applied at very high doses per patient, these numbers actually multiply very quickly by a factor of 1,000,000 or more, which means the classical production methods are really not applicable to meet market demand, and scalable processes are required. And this is what we are targeting with our ELIVector platform, which we developed exactly with the goal to have a production method for AAV vectors for gene therapy that can be scaled like you know it from processes for antibody production, for example. And how does that work? On paper, it's very easy. It took us a bit more of an effort. It wasn't that easy, but we made it. So the method we are applying can work independent of any plasmid transfection. So you get rid of the whole triple transfection process of expensive plasmids, of expensive transfection reagents, often not so well controllable, not so controllable process. The method also, unlike some other methods, does not lead any helper virus infection. So what we're doing with the ELIVector platform is we're actually generating truly stable cell lines that carry all the components required for a producer cell to make AAV vectors. And these stable cell lines, as you know it from antibodies, from recombinant proteins, can be banked, can be GMP banked, and then expanded up to the production volume. Production is induced then, and the AAV production process starts. And I'll show you some data on how that actually works. Just to basically give you an idea on how it applies in real life. Obviously, for an AAV vector, you always need a product-specific gene of interest, a therapeutic gene that's delivered as a cargo. And also, in many cases, a specific capsid is desired. So it can be one of the common, well-known AAV serotypes like AAV2, 8, 9, but very often our partners also have developed their own proprietary serotypes, which they would like or which they are using in their AAV therapeutics. So what we have created is a starting point, our alpha cell line, which carries the rep and helper genes, which are required for AAV production, stably integrated. And this serves then as the starting point to stably integrate the product-specific capsid and the product-specific therapeutic gene to generate the producer cell line. And from such a producer cell line then is very easy to produce, and we have actually shown that in a proof of concept experiment with the proof of concept cell line that I'm showing you here. So we initially generated a polyclonal producer pool. That means this is still a polyclonal cell population carrying the gene of interest and the capsid, both being polyclonal for both of these. And when we look at production from such a pool, we already got decent titers in the 10 to the 13th range, either in an embryo system or in a 10-liter stirring bioreactor. When we then go through single cell cloning, what we and screen with an embryo 15 stirring system, stirring bioreactor, we gain an order of magnitude in productivity, reaching 10 to the 14th viral genomes per liter without any process optimization in this particular instance. And of course, we then really wanted to show and demonstrate the scalability. So we worked in a collaboration with Paul, and they used their Allegro stirring bioreactors initially at a 50-liter scale and then scaling up to 200. The scale up was very smooth and easy, going from 10 liters to 50 to 200. And as you can see, actually in the growth data, is that we see very comparable growth curves. We see very comparable growth curves in the 50-liter and the 250-liter runs shown here, which is the blue lines, and compared to the 200-liter run, which is the green line. Basically, the cells grow up to about 5 times 10 to or 5 million cells per mil in 3 days, and then we induce our collaborators at PAL, I should say, induce AAV production. And as you can see, and as is expected and known, from there on, the cell numbers stayed constant and started also, along with viability, to slowly decrease, which is known, a known effect of the AAV production of the cells. When we then look at titers, we see the same high titers that we had in the 10-liter bioreactor run. So we're close to 10E14 viral genomes per liter, and without any drop in the 200-liter runs. And I'm showing you here day 4, 5, and 6 of the stirring bioreactor runs. And if anything, the 200-liter yields are slightly above the 50-liter yields, but there is definitely no drop in productivity here. So, of course, we were already very excited about these data, but it's not the end what you can do, because this is still not non-optimized stirring bioreactor process. It's nicely scalable. We don't have any doubt it will easily go beyond the 200 liters, which is sort of a critical volume for upscaling. But of course, there's more you can do to a stable cell line. And this is what we did in the next step. And what we did is we actually applied perfusion process. So this actually helps to increase the initial cell density and also to improve the process itself. So what we did here is we let the cells again grow in and this was a smaller scale experiment just to prove that it works. We grew the cells at 1 liter scale bioreactor for, in this particular case, 3 days. Then started the perfusion process which basically increased the viable cell density to 20 million cells per mil. So we had a 4-fold higher cell density to start with, and then induced again AAV production, and then looked at AAV titers basically for the next 5 days. And what you can see is that although we only increased the cell numbers by a factor of 4, we got a 30-fold increase in titers. So we saw up to 3 times 10e15 Vg per liter, which means we see about 10-fold higher cell-specific yield in this process. And also we get very good full empty ratio. So when we look at the percent of full particles, we are between 30 and 40% full particles in this process. So we have a method in our hand that gives us high titers, good quality AAV particles, and which is amenable to upscaling if you're going with bigger bioreactors, and there's 1,000-liter perfusion processes actually established with many of the CMOs. We have initially established the Elevector platform on our amniocyte cell line, the CAP cells, because we were very confident we could get it to work. I mean, it all looks easy, but it wasn't trivial. A lot of people predicted we couldn't get it to work. We managed to establish this platform on the CAP cells, but we have also got, we're getting requests to have it also on a more widely used cell line, like the HEK293 cell, which is the standard cell line for viral vector production. And so this was the next step then for us to transfer and to duplicate the platform on HEK293. So the first thing we did is actually generate our own trusted 293 suspension cell line. So we used the adherent 293 cell from ATCC. We adapted them to suspension in animal component-free, chemically defined media, which is of course the required state of the art. We then did a subcloning of this suspension HEK cell line and tested these subclones. And I'm just showing you basically 2 of the subclones here that were just tested for the ability to generate AAV by triple transfection, so the standard triple transfection method. When we compare them to commercially available 293 cells, and these are high-performing cell lines of competitors, we see the same titers in our 293 cells. So we felt confident to move ahead and actually duplicate the Elevector platform on these cells. And we have got alpha cell lines on 293 cells. We have got the first proof of principle data, in this case actually from a polyclonal population that's polyclonal for all 4 genes. We can already detect decent titers in these cells, so we feel very confident that it will be possible to get high producing producer cells from this platform once we do the subcloning. And the platform is well advanced, including the alpha cells. So we will be offering them to our partners in the second half of 2021, so the second half of this year. How do we actually work with partners? So we are not a drug development company. We're really specialized and focused on developing and the technologies and making them available to the gene therapy companies. So we're offering it to our partners. We will also do, if in most cases, the cell line development for the specific producer cells, and then do the technology transfer and out-license the technology and the cell lines to our clients. And as you might have seen, we have ongoing collaborations with a number of companies, for example, with Roche, Spark, and Biogen. And at this point, I would like to thank you for listening to this talk. And if you have any question, I think there will be a box or a chat where you can leave your questions. So thank you very much.